It's great to welcome to the program today Vincent Racaniello, who is a virology professor at Columbia University and also host of the podcast This Week in Virology. It's so great to have you on today. I appreciate it.
My pleasure. Glad I could make it after all that rain yesterday.
Yeah, I saw the videos. It's something else. Listen, I'll just jump into some things because there are maybe some specific things that are new ground for my audience that I think might be interesting to talk about, and I think it'll open up a broader discussion with something like the idea that there's a period of time during which you can be incubating this virus. In other words, you have been infected. You don't yet have symptoms. You would not yet test positive if you were to be tested. What is actually happening during that period of time? Is it that there are some virus particles in you, but they've not established themselves in your system? Like, what is taking place at that point?
So let's think when you get infected, you're typically inhaling droplets that have been produced by someone else, right? They're infected, their droplets have virus. You inhale them into your nose. Typically, they go into the cells lining your nose and further back, which we call the nasopharynx. And the viruses then attach to cells. It's not easy to do that. That's why a lot of viruses have to get in you to start an infection. But the viruses attach to cells, they're taken into the cells, and then they begin to multiply. And in the very first hours, it's very low levels. The viruses are in maybe a few cells. They're beginning to multiply. You can't really detect it by PCR. But then you go above a threshold where those cells produce virus. The virus infects neighboring cells, and it goes on and on. Maybe it takes about 20 hours for a cycle to complete in a cell. So after a few days, you are now at the point where you would test positive by PCR. So in that interval that you're interested in, the virus is multiplying, your immune system is beginning to detect it. You have the earliest immune system, which is called innate immunity, can detect what's going on there. And in fact, most infections are stopped very early on before they progress any farther. But then if the innate immune system doesn't work, and part of that is interferons, which you may have heard about, which block virus infections in many ways. If the innate immune system doesn't work, the virus keeps multiplying, and then we have activation of other immune systems, which include antibodies and T cells, and then it becomes a race to see if the virus can outpace it. And then after a certain number of days, maybe 5 days on average, you might then feel symptoms. You might get a scratchy throat, you might get a headache, you might get a fever and so forth, which might prompt you to go get tested. And bingo, there you go. You get a PCR test and you're positive.
I have friends who have gotten a bunch of the Abbott home tests. And for example, a friend had a birthday party with 10 people last week. And of course, everyone had to be vaccinated, but he also tested everybody on the way in. And his belief was, if there was a negative from someone who was in this state you and I just described, the virus has entered their body, but they are not yet testing positive, that it's okay because they are almost certainly not contagious at that point. Is that actually our best assessment of one's level of how contagious you are during that intervening period?
Yes, the PCR tests are very sensitive. So if they are picking up nucleic acid, you have a good amount of viral multiplication. And if you're negative on this test and you go to an affair for a couple of hours, certainly in the time of that, you're not going to change from non-infectious to being infectious. So if you're negative, I would say you have 24-hour window before you might even start shedding virus and infecting others.
When it comes to the conversation of variants, I've read a number of pieces that say that evolutionarily, the way that there is selection pressure on these variants, the idea of a significantly more deadly variant becoming dominant is not particularly likely. I know that there's a lot in there about level of infectivity versus the deadly nature and a lot of things to unpack. But let's start with, is it true that it is unlikely that a much more deadly strain would become dominant? If so, why? If not, why not?
When we look at all the viral outbreaks in humans over the years, for the most part, viruses do not become more deadly. If anything, over a long period, they become less deadly. They become better adapted to their hosts. And I'm talking many, many years, maybe hundreds of years in time. And so there's no evolutionary reason why a virus would become more deadly, because it would kill its host faster and it would be less likely to transmit. And transmission, of course, is the raison d'etre for a virus. So the variants are certainly arising because of some kind of selection pressure, right? We think part of it is immunity in the population. We didn't see these variants until late in 2020, when a lot of people globally had already been infected or vaccinated and had some kind of immunity. So antibodies can certainly put pressure on a virus so that you would select a virus that is slightly different and can evade the antibody. It happens with influenza virus every year. And there are probably other selection mechanisms that we don't know about, because these variants have a lot of different changes. So I think in the end, what we can say about the variants, they're selected probably mainly because of immune pressure. They're not likely to be more deadly, even though there's some studies here and there that suggest that they're not very well done. They're not very well controlled. I think the main issue is immunity and escape from it.
And over a period of time, would we, or let me put it a different way, there is already a trial going on with a modified mRNA vaccine tailored to more recent variants. I don't know the exact timeline of when that may become the one that you get when you go to CVS or Walgreens or wherever the case may be. Is it likely that, like the flu shot, we would need to be tailoring the vaccine to the most recent variants?
It's certainly possible. Now, with influenza, that can happen every year or two, right? We tailor a new vaccine. And we have so much experience with influenza that we know when antibody levels drop below a certain point in people, we know that correlates with them getting more severe disease. We don't have that information yet for SARS-CoV-2. We don't know what antibody levels are protective. We don't know how low you can go and still be protected from disease. Right now, the vaccines protect against severe disease and death, even though antibody levels are declining. So it's not clear when we would need to change the vaccine. I think we have to wait and see if more and more vaccinated people, this is the key, do fully vaccinated people start to get more sick? Do they get severe disease? Do they die? When we reach that point, then we talk about changing the virus vaccine.
Earlier on, there was the idea that was much repeated, I guess we would call it a belief, that the 2-dose regimen of either the Pfizer or Moderna vaccine would likely be good for at least a year. There were discussions of 2 to 3 years, et cetera. More recently, there's been the data from Israel that shows that the antibody levels, although, as you say, we don't know exactly what that means in the real world, but antibody levels seem to start declining after 8 months. There's the idea maybe after 6 months, one would get a third dose. I've also read some doctors say the third dose will almost certainly be the last one. At this point, it seems to me we really just don't know. We really just can't say 3 is going to be it. What is it that we need to be looking at to really get an answer to that question?
So one thing we have to understand is that with every human viral vaccine, you get vaccinated, 6 to 8 months later, your antibody levels decline. It's the natural process. There's nothing new about SARS-CoV-2 vaccines and antibodies declining. I mean, although people are making it out to be a big deal, they are declining. And with that, there has been a push for boosters. What's not declining, though, is protection against severe disease and death. That remains robust. So the antibodies level decline. You may get infected even if you're fully vaccinated, but that happens with just about every other human viral vaccine that we know about, but you don't get serious disease. That's how we tested these vaccines to prevent COVID of any kind. Now we're getting mild COVID even after fully vaccinated. And so the key for a booster, so I think the booster idea is somewhat flawed, because the idea is to push antibody levels up again, but they're going to decline in 6 to 8 months. So are we going to go every year and give people a booster? I don't think that's a good public health strategy, frankly. The key is, do we still prevent severe disease and death? So far, we are doing that. And as long as that protection remains strong, then I don't think we need to have a booster. And to think that it would be the last one, well, there's simply no data to suggest that. I think that would be great if it were,
Yeah.
but we simply don't know.
When it comes to the seasonality of this, when we were going into last fall, there was, generally speaking, a consensus that we were going to see a big increase, and the explanations varied depending on what you looked at. Part of it was a similar explanation with influenza, that as it gets colder, people spend more time inside, and that's going to increase transmission. We were not yet vaccinating at that time. This fall, we seem to be seeing at least the growth of cases slow down. It's not yet declining, but if we're 6 to 8 weeks after where Europe is, maybe we are starting to get to a peak. So maybe we won't see that increase into fall and winter, although maybe we will. What's our best assessment right now as to the quote seasonality of this, and what is actually the source of that? What generates that?
So we really don't know how seasonal COVID is, right? Because it's brand new. And when you have a pandemic, that trumps all seasonality effects. So, for example, in 2009, the pandemic influenza virus emerged in August, which is totally weird because they usually come out in the winter. But when there's a pandemic, most people are not immune, so the virus can spread even though it's the wrong season. So that's what we're seeing with SARS-CoV-2. Now, there are other coronaviruses that infect us. They cause common colds. There are 4 different ones. They are seasonal. And it's likely just by extension that SARS-CoV-2 will be seasonal as well. Although still we're in this pandemic phase where seasonality gets trumped by simply, if you have a lot of susceptible people around, which we still do, the virus will reproduce whenever it wants to.
What is the actual mechanism for the seasonality? Is it as simple as the temperature forces people indoors and increases spread?
Well, that's a good question because seasonality is different for different viruses. In the heyday of polio in the world, that was one that spread in the summer, right? Warm weather when kids were out playing and so forth. But influenza is in the winter in seasonal in temperate climates. And we think that's because the low temperature allows the virus to remain infectious longer, and the low humidity keeps those droplets that you exhale, keeps them very small, because in humid conditions, they take on water, they drop to the ground more quickly. When it's dry, they can travel farther and infect people. But we don't know much about that for most other viruses. It's a really tough topic to understand.
So I know how to frame my next question. Have you seen the mask efficacy study from Bangladeshi data?
I have not.
You haven't? Okay.
Tell me.
So my understanding of the Bangladeshi study on mask efficacy is that for older cohorts, there was definitively reduced transmission from mask wearing, although the statistical significance of the reduction seemed to be lower the younger the cohort you looked at. There may be other confounding factors there. So, I think that the devil is probably in the details. What's our best assessment right now of the impact of masks on reducing spread? Because it's become very politicized and controversial, and maybe the science doesn't suggest it should be.
Yeah, so masking is tough because it's hard to do a good study, right? You would love to be able to take a group of people with masks and without and challenge them with virus and see how the mask, but we just can't do that. So we have to use surrogates. And so I think the best estimate for COVID is that when worn properly, and that's a big if, because not everyone wears them properly. And I see more and more people just having them below their nose because they're so used to it. Maybe it stops 60% of transmission. And it makes sense because the droplets that carry the virus, they get caught up in the mask material. It's not the virus that gets trapped. It's the droplets carrying the virus. So if you wear them properly, so there are no gaps, maybe 60%. It's not going to be 100% ever. And we know that in many countries where they use masks quite often in respiratory virus season, it does make an impact. So and my view is that it's an easy thing to do. It's not too much of an inconvenience. I'm used to it by now, so I don't really see the issue.
When it comes to treatments, I'm glad to have you weigh in on particular treatments if you feel so inclined and as though you can do so. But more generally speaking, what is the reason why some of these widely available cheap drugs enter the news cycle as possibly efficacious treatments? Is it simply that there's stuff lying around that's cheap and doctors decide to throw it at the virus and see what happens?
Yeah, I think that's part of it. I mean, the tendency is to take a drug that's already approved, say by the FDA and licensed, and you can use it off-label in people without too much trouble. So for a few drugs early on in the pandemic, some labs did experiments showing that the drugs could inhibit virus multiplication in cells. And that was immediately translated to usage in people without a good clinical trial. And so none of those repurposing have really resulted in anything worthwhile. It's unfortunate because you would love to have a pill that you could take at the moment of your positive that would help you a lot. We still don't have that. And if you want to know why, besides these repurposings failing, it's because the billions of dollars were put into vaccines rather than drugs. And that's the bottom line. If we put $20 billion into drugs, we'd have some right now.
Is that right? You're confident that this is not an unsolvable problem. It's just not been, we haven't put enough into it yet.
Oh, absolutely. And in fact, I think we could have had a drug before the pandemic even started. We could have had one ready and it would have stopped the pandemic. And that's a failing of research and big pharma for sure.
What type would it be? An antiviral, or what type of drug would it be?
It would be an antiviral because we knew after SARS-1 that there were similar viruses in bats that could infect human cells. And all we needed to do was pick a target, which is highly conserved among those different SARS-like coronaviruses in bats, and develop an inhibitor. Easy, could be done within a few years, not very great cost. It could even be tested in a phase 1 and be ready for subsequent studies once the pandemic began.
When it comes to the hypotheses about the origins of this pandemic and this virus, there was a period very early on where so-called lab leak hypotheses made a splash. It sort of went away. A few months ago, it was back. Joe Biden was asked about it. Jen Psaki was asked about it. Basically, everybody said, we should investigate everything, but until we have evidence to believe something, we shouldn't say that that's what's most likely. It's now disappeared again, essentially, from the public discourse. To me, it seemed very sort of media driven. There was a decision made implicitly that now we're going to ask about this, and then it sort of went away. Where is your mind on how this should be investigated and whether there's any there?
It's always been clear to me that this virus, just like SARS-1 before it and all the other coronas, and in fact, all the human viruses, it came from a wild animal somewhere, not from a lab. No virus that has spread this way has ever come from a lab, which is not to say it couldn't happen again, but that lab in Wuhan was not working on these viruses. There's plenty of evidence that they're closely related viruses in nature. And I do think that the lab leak idea is just a distraction, politically motivated. It's an easy answer for a tough problem. It took 10 years to find the virus which came into people for SARS-1. And so it might take as long to find where that virus is, the precursor. We don't have anything close to it yet. It requires a lot of wildlife sampling. And if we're going to have international arguments with countries, that's not going to happen. But a paper just came out last week showing that if you look at rodents in China, you can find these coronaviruses. So we've barely sampled wild animals, and that's where the answer is going to be for sure.
Last thing I want to talk about, there was the likely imprudent idea of Easter 2020 as when things would go back to normal from the former president. That was not particularly realistic. There were high hopes for this summer. Dr. Fauci has recently talked about spring of 2022 if there's enough vaccination. Do we really know when the serious decline of the pandemic is going to be at this point? And does it simply depend on variants and vaccination?
Well, I was telling people in the spring that by this fall, 2021, it would be controlled in countries that vaccinate 80% of the population.
Right.
And unfortunately, we haven't reached that in the US, so we're not controlling it. And I think if we get to that level of vaccination, you will control serious disease. It doesn't look like we're going to get there in the US. We might have to get there by some natural infection, which is going to take longer because every cycle of outbreaks doesn't get everyone infected, and you have to depend on another one. If we could vaccinate 80% of the population by the spring of 2022, then yes, the US could control it. But of course, many other countries will not be able to get there. So we're talking about a few more years globally until we get most of the human population vaccinated.
I'm trying to do the math in the US. If we're at 52 and we want to get to 80, that's 28 additional percentage points. We're at the beginning of September. So you're talking about, if we say spring is the end of March, it's 7 months to do 28%. It's only 4% a month, which seems so doable. And yet we see it move by 1/10% a week. And then it's not looking particularly good.
Yeah, there are a couple of problems. Of course, one is that under 12, you can't be vaccinated yet.
Right.
And we're hoping that changes by the end of the year. That would make a big difference because, if you had 80% of the population immunized, you would still have classrooms with 30 kids with no vaccine. That's a problem.
Yeah.
No one's going to be protected.
Yep.
And then, of course, there are people who simply don't want to be vaccinated for various reasons, and it's unfortunate. And I try and reason with them calmly. I'd love to talk to them one-on-one, but some of them are just dead set against it. So that's the obstacle.
Yeah, that's for sure. Make sure to check out the podcast This Week in Virology. We've been speaking with Vincent Racaniello, who is not only one of the hosts, but also a virology professor at Columbia University. So great having you on today.
My pleasure. Great conversation. I appreciate it.